Uh, Good morning, Mr. Schekman. For me, it's a pleasure to speak with you. I am first journalist from Moldova who take interview from you.
Uh-huh. Well, yes, you are. Ian is from Russia or from the Soviet Union, so maybe not. He's not Moldovan.
Please tell us about your grandparents. We know you have Basarabian roots.
Yeah. So my grandparents on my mother's side were from a village called Lipcani.
Yes.
And as far as I can tell, their parents, or my grandfather's parents, had come from Ukraine and moved. My great-grandmother, I believe, came from a family of some means. I think they owned a brewery, and they moved to Lipcani. I don't know the circumstances of their moving, but then they died in the early 1920s, my great-grandparents, and are buried there in the Jewish cemetery. And when I first was in touch with Ian of the Moldovan Academy, he had a representative of his academy living near Lipcani who visited the village and sent back this wonderful picture of the Jewish cemetery with all of the tombstones all sort of in disarray. It's obviously been abandoned and not maintained. And so that was a haunting image to see these tombstones like this with the the Yiddish print on the tombstone. So I have resolved, when I finally come to visit, I hope next year, that I will make a journey to Lipcani, and I have the written impression of what my great-grandparents' name would have appeared like, and I'll look in that cemetery to see if I can find their gravestone. In any case, I will have been the first member of my family to return to that village in 100 years. So no one has ever gone back. My grandfather was a tailor in the Romanian army at the time, and for reasons that I don't understand, I didn't hear about, they entered a lottery to emigrate from Basarabia in 1927, which of course was most fortunate that they got out. Unfortunately, they emigrated to Germany in 1927. I think they recognized that was a big mistake. So they somehow escaped, and they got on a boat that came to America. They landed not at Ellis Island. All of the famous stories of immigrants who come from Europe to America, all of them seem to land at this famous place in the New York harbor called Ellis Island, where there's a great monument. It's a national park. But my grandparents on my mother's side somehow went to Providence, Rhode Island. I don't know why. And they went to travel to New York, but my grandmother didn't like the big city. And they had a kind of an adopted cousin whose family had moved from Lipcani earlier to settle in Minnesota, in Minneapolis, Minnesota, where my grandparents then moved, and they settled down. The Depression set in, and so it was a very challenging time for them. My grandmother contracted tuberculosis during the Depression and had to go into a nursing home. And my mother and her sister, who were born in the late 1920s, had to be in an orphanage for a couple of years while my grandfather struggled to make a living and to support his wife in this nursing home. But they eventually recovered, and my mother grew up in Minneapolis and met my father, who was from another Jewish community in the neighboring city of St. Paul. And then we settled down. I was born in the late 1940s. And for the next 10 years, we lived in Minnesota before we moved to California. So my family life in my early years was always centered around my grandparents on my mother's side. Every Friday evening we had a Sabbath dinner at their home. We would go to the synagogue with them, and the life revolved around the family. That area of Minneapolis, Minnesota is known for the Scandinavian immigrants, Swedes. But I never knew any Swedes because everybody in my community was Jewish. I thought Minnesota was all Jewish. So I lived in a Jewish ghetto, basically. And it wasn't until we moved to California that I realized that Minnesota was mostly Scandinavians. So anyway, my grandparents, of course, they had a very difficult life in Basarabia. I think the prejudice against the Jews was really enormous, and they somehow survived. And they wanted to escape. And my grandfather, years later, would say he would never go back, even to visit. He has such bad memories of the way the Jews were treated. So that's my knowledge about about my ancestors, and I'm sure. And then subsequently, after they left, there were some other members, I think sisters of his, who moved to Israel, but everyone else in the family was slaughtered by the Nazis, and so there's no one left. No one left there.
Why did they choose to go in the United States or not in Canada or Latin America?
Yeah, well, I think it was because they had some distant family in the United States. I'm not sure why they didn't choose Canada. They had some family, not many family members, but some, and they thought, well, this was the freedom of religion in the United States, I'm sure, drew them. I'm sure it was primarily the freedom of religion and the opportunity to have a new life. So they were adventuresome in that way.
Did the grandparents also speak Romanian or Russian?
I think they just spoke Yiddish, basically. They may have spoken some Romanian, but mainly Yiddish.
Only English. Only your maternal sides have Basarabian roots. I understand, right?
Yeah, my grandfather on my father's side came probably from Russia. I'm not sure. I have much less information about that. He came to New York in the early teens, 19. Actually, his older brother had emigrated to Massachusetts, where my father's grandfather first moved. But then he ended up in New York City by himself as a teenager, and he enlisted in the British Allenby Brigade that fought in Palestine before World War II, and so he was a soldier in Palestine until he returned to the U.S. and somehow ended up in Minnesota. His wife, my grandmother, died at a young age. I never met her, and I think her family must have come from Russia. My grandmother on my father's side, she had a very large family, something like 9 children, and they represent a very large number of people who are spread all over the world. Many are in California, and one of them presided over my bar mitzvah in Los Angeles. One of the relatives of my father's mother. So I knew some of these family members, but not well. And they didn't come from Basarabia, I don't think.
How have they adapted to the United States since two different cultures and languages?
Yeah. Well, I know they struggled, and they probably gathered in communities where they could speak Yiddish with their close friends. And my grandfather on my mother's side, the one from Lipcani, learned to be a tailor, and that was a trade that he took with him to Minnesota. And he was, reasonably middle class, successful as a tailor, and he would make clothing for us. My suits, my first suits when I was growing up were from my grandfather. His claim to fame was he once tailored a coat for the man who would eventually become vice president of the country, Hubert Humphrey. So he was honored to be able to make a coat, a winter coat for Humphrey, who was then the senator from Minnesota. So that was his claim to fame. And on my father's side, my grandfather on my father's side was what we call a jack of all trades. He would do lots of things. He owned property. He was a traveling salesman. He did all kinds of things. And from my father, I gather he was fairly prosperous in that Jewish community in St. Paul, and likely, in fact, was always, giving money to family members to help them out. So he had money, but I didn't know him as well as my grandparents on my mother's side, but because his wife had died before I was born, and he remarried and then moved to Florida, away from Minnesota when I was still young. But I met him in Florida on one occasion when I was a teenager.
Some holidays from Basarabia or food traditional.
I think again, everything culturally that I experienced as a youth was related to our religion, and not I don't think, not that I'm aware of anything that came from Basarabia or Romania. It was all Jewish tradition, so Jewish holidays were what we celebrated. So I think the Jewish community in Basarabia was probably very insular, isolated, culturally segregated. Lipcani at the time may have been almost entirely Jewish. I just don't know. There's probably some history about Lipcani, which you may know more about than I do.
In Basarabia existed 18 big communities, agricultural colonies, we can say, where in Lipcani it's big community. It's maybe 80% of population was Jewish.
Yeah.
You can speak about your parents?
Sure. So my parents were both born in Minnesota, and my mother in Minneapolis, my father in St. Paul. They also grew up in a isolated, segregated Jewish community, and they met when my mother was in high school, and they married. My father was drafted near the end of World War II, and he had to go to military training, but he never left the United States. He didn't have to go to war. He was just a little too young. And so my first memory of them was when I was maybe 2 years old. We lived in Minneapolis in an apartment. And when I was 10 years old, my father answered an advertisement for a job in radio technology in Los Angeles. And now he had spent his entire life in Minnesota, so he knew nothing else. But it was the middle of winter when he went for his interview in Los Angeles. And he was so amazed at the nice weather that when he had the job offer, he came back home to Minneapolis, and he basically said, "We're leaving, we're out of here." And we drove in the middle of winter, late December 1959. We drove all the way from Minneapolis to Los Angeles, and we were there New Year's Eve, 1959. And he loved California, and he hated to go back to the winters. He never enjoyed travel back to Minnesota. My mother, on the other hand, missed her parents, the Basarabian grandparents, missed them so much that she was miserable for years. She hated California. She wanted to be home with her parents. But they would come and visit very frequently. So we remained very close to her parents, my grandparents. And so an amazing coincidence, many years later, when I was at a meeting of an honorific academic society called the American Academy of Arts and Sciences. And I was one of the officers of that organization. And I had the pleasure of introducing new members, one of whom was someone from Columbia University, whom I knew because he was in my general area of research. And he had years earlier told me an amazing story. He said that my mother and her sister, when they were in the orphanage, because my grandmother had tuberculosis, his mother had an older, who a few years older, had also been in this orphanage and befriended my mother. This was in the early 1930s. I mean, amazing. And this woman, who was still alive at this point, she was in her 90s, recalled my mother from that time. My mother had died already by the time I met this woman. My mother had a brain tumor and she died in her late 60s. And so to meet this woman and to learn of that connection to my mother was very emotional. So
I read the history with microscope.
yes. Yeah, so that was, of course, I think that'll end up on my tombstone. So yeah, that was, I was, when I was a child, I was single-minded in my interest in microorganisms. Where this came from, I have no idea. But anyway, so, I mean, you read the story. So I had this toy microscope and I wanted to buy, a serious microscope. And so I saved my money and my mother kept borrowing the money.
I know.
So I had to run away to the, yes.
Sorry,
yes,
I know this
that.
history, but other people not say. Please say about this.
Okay, sure. Okay, I'll say about it. Yeah, so when I was maybe 11 years old, I had a toy microscope, just a little plastic microscope. And I collected a jar of pond water from a local creek. And I took a drop of this and I put it on the glass slide and I looked in the lens and I saw this world of microorganisms swimming, crawling. I mean, it was unbelievable. These things you can't see with your naked eye, but just with a little bit of magnification, you can see a world that's as complicated as what we see with our eyes. And it was just, for me, endlessly fascinating. I would just look and watch and try to imagine what was going on there. And so, but I realized this was only a toy. And one evening, I was describing this to my family at dinner, and my father was sort of dismissive. It's just a toy. What can you see in a toy? And so I decided at that point, okay, I'm gonna save my money and get a real microscope. So I used to do lots of chores. I would mow lawns. I would babysit for neighbors. I would deliver newspapers. And I collected money and I put it in an envelope in the closet in my bedroom with a goal in 1962 of getting $100, which was a lot of money. And I could never get to the $100 because my mother would borrow money from the closet. And I don't know why she, didn't pay me back. One Saturday when I was mowing a neighbor's lawn, I was so upset that I bicycled not home, but to the police station. And I told the duty officer that I was running away from home because my mother was stealing my money and I couldn't get my microscope. So they called my father in to the police station and he was quite angry. But he went into the captain's office and they had a few words and he came out. And I learned a valuable life lesson that afternoon because we drove right to a secondhand store in a nearby city. And there was that microscope. For $100, it was a student professional Bosch & Lomb microscope with, serious optics. And so that became my pride and joy for the rest of my time that I lived at home. Every year, I would use the microscope to study blood cells or microorganisms for a science fair project that I would prepare for display at the school science fair and then, into the county science fair and then finally into the California State Science Fair. And my best achievement in my last year of high school was I took fifth place in the high school biology division. That was my biggest achievement. But that comes full circle, and I'll tell you another story you probably don't know. So then I went away to college, and the microscope got put away in the garage of my family home. And years later, when I became a faculty member at Berkeley, my parents sent the microscope up to my new home. And neither of my children ever showed any interest in science. So the microscope just sat in the closet and collected dust. And then when I won the Nobel, a month or two later, I got an email. Every year, the Nobel Museum in Stockholm sends out a request to laureate to send in something that reflected their discovery in science. And I didn't have anything nearly as dramatic as that microscope. So I dusted it off and sent it to Stockholm. And you can go, and it's still on display. I'm told it's a very popular exhibit because there's a caption underneath in English and in Swedish that says how I had to run away from home to begin my career in science. Now, after the Nobel, and here's a new story, I got an invitation from the California State Science Fair, where I had done, exhibits when I was in high school, including winning 5th prize. And so the gentleman who introduced me to all the high school students from the state who were showing their exhibits for the state fair gave an introduction where he talked about my Nobel Prize and my discoveries. And then he said, but the most important thing he did in his life was to win 5th place in the high school science fair in California, because now those of you who did not win 1st place should be encouraged that 5th place isn't so bad. So that was fun.
How do you spend your time in childhood? Maybe you have a hobby?
Well, my hobby was science. I mean, working on science fair. I played, Little League baseball, but I was not at all athletic. I read a lot. I spent any time that I wasn't in my bedroom looking at my microscope, I spent in libraries trying to dig up, science journals. I remember I went to the local library and I asked for Science Magazine, which the library had. And the librarian said, you can't have Science Magazine, you're just in high school. And so that was ridiculous. I would go to bookstores and I would look for old science books. I mean, I just was, totally absorbed by science. And I remember reading about the various colleges that I might go to. Berkeley was, of course, one college that I considered. And I remember looking through the catalog for Berkeley, and some of the professors at Berkeley at the time had written books that I had read on microorganisms and molecular biology. But it was farther away from home, and money was always a major issue. My family was not rich. And so I went to UCLA because it was closer to home and less expensive. And so I feel, very strongly about public higher education. I'm a product of the University of California. And so for me, that's really made my life.
Please tell us about the history with great immigration to California.
With
Great
immigration to California?
To California. From your hometown to California.
When we moved. Well, it was the winter of 1959. It was snowing. It was treacherous. The roads were icy. And at one point, my father put on the brake and the car spun around 360 degrees on the ice. It was kind of scary. And again, we didn't have much money, so we stayed in not so nice hotels with very little heating. And we arrived late to Los Angeles. We missed the reservation that my father had made at a motel where we were to stay until they found a place to live. And so the only thing available to us as a family, we had 4 children and 2 adults, was an unheated room with 1 bed where we all stayed in the bed together to stay warm. So that was welcome to California, where it was cold in the winter. Once the other motel opened up, we stayed there for several weeks until my father found a rental home in the San Fernando Valley, where we stayed for the next year until we moved to Orange County, where my father bought a home in a nice neighborhood. And we lived there. And my father stayed there until my mother died for some 40 years. And then he remarried and moved to another home. My father died at age 93 last year. But fortunately, he was there, still alive when I won the Nobel. And every year before I won the Nobel, he was eagerly awaiting that early October because he was sure that I was going to win the Nobel. And because I had won other prizes that were sort of, precursors of the Nobel. And so on the very morning that I won, he was the first person I called. It was 1:30 in the morning, because when they call from Stockholm, they call as soon as they make the decision. And in Stockholm, it's mid-morning, but in California, it's, in the middle of the night. So when the phone rang at 1:20, my wife, who was quite ill already at that point, heard the phone ring first, and she yelled out, "This is it," somehow anticipating that this was the call from Stockholm. So I got the phone, and I was very nervous, and I got the news. I called my father, and his wife answered the phone, and she started screaming. And my father was really taken aback because he had forgotten it was that time of year. And so finally he got his good news. And for the 2 months after the announcement of the prize, before we went to Stockholm for the prize ceremony, every time some salesperson would call the house to try to sell my father something, he would say, "I'm not interested in what you're selling, but let me tell you about my son." And so in Stockholm, there were many events, but one event that I'll, of course, remember is you have to give a formal lecture to the Karolinska Institute, a medical school. They had a new auditorium that held 1,000 people. And so Rothman and Sudhoff and I gave lectures. And when I gave my lecture, I made my father stand from the audience and to receive my appreciation. And I congratulated him on surviving to see this day finally.
Please tell us what was about your first experiment in biology?
My first experiment in biology, well, those were experiments, sort of experiments that I did in high school with microorganisms. But, I did not, my high school biology teacher, who took an interest in me throughout my years in high school, he was not an experimentalist, so he wasn't much help. So I didn't have a real scientific mentor until I got to university. So those experiments that I did, looking back, they were not well controlled. I wouldn't trust them. The first experiments that I did that really worked were when I was in university. So when I left high school, I left with the intention of going to medical school. In the US, unlike in the rest of the world, you first go to university for 4 years to get a degree, bachelor's degree. And then if you want, then you can apply to, and if you've done well, you can go to medical school. Unlike, in Europe, where you apply out of gymnasium, for instance, you apply directly to a medical curriculum. You don't do that in the US. So when I started at UCLA as an undergraduate, it was with the intention of eventually going to medical school, like many of my fellow students who were interested in biology. I took first year chemistry from a really wonderful teacher, just a wonderful lecturer. And I did very well in the class. And I was put in a special honors section. And in this honors section, in addition to having lectures from a Nobel laureate, actually at the time, every student in that section was assigned or selected a chemistry professor's laboratory to work in for a couple of months. And so the first experiment that I think I really did carefully was in this laboratory, chemistry laboratory, when I was a first year university student. And it was just a simple experiment where I took a sample of DNA and I broke it up into the bases using acid hydrolysis. And then I separated the bases or nucleotides on a paper chromatogram, and then just saw the peaks of UV absorbance to reflect the migration of these different nucleotides. It was a trivial experiment, but to me, it was it really resonated with the way I had been thinking independently anyway. And so at that point, I decided, no, I'm not going to medical school. I'm going to be a scientist. So I had to tell my parents that summer that their son was not going to be a real doctor. And they were disappointed, of course. And especially my grandfather from Basarabia, very disappointed that I wasn't going to be a doctor. But they eventually got over it. So
Please tell about your relationship with Professor Jack Hoskins.
Yeah, Jack, he was my teacher in high school. Yeah. So I met him when I was still in the school before that, which is called junior high school. I was in 9th grade. So I was 14 maybe. And I was at the county science fair where I'd done my science project. And he came up to me and he said, "Do you know who I am?" And I didn't know. I thought he was a friend of my parents. He said, "No, I'm Mr. Hoskins, and I'll be your biology teacher next year." That was very nice of him. And so I got to high school, and sure enough, he was not only my biology teacher, but he was my teacher in different courses every year. And he took really a direct interest in nurturing my curiosity. Although he didn't know how to do experiments himself. He liked plants. He grew plants. So he was a sort of a systematic biologist, but not an experimentalist. But he was very encouraging, and he kept in touch with me after I left high school. For years, he would contact me somehow. He came to visit my laboratory when I became a professor at Berkeley. And then on the morning of the Nobel Prize announcement, he sent me an email to say that he was so proud to be able to live to see that day. So anyway, then after that, I did a return visit to my high school. I hadn't been back in 50 years. And I was the kind of social misfit, outcast in high school because I was a science nerd. I was not socially adept. So, but when I came back as a Nobel laureate, I was returning as the conquering hero. I drove up in a limousine. I was greeted by the cheerleaders and the marching band and all the dignitaries from the school district. There were big posters all over the school. So life has surprising turns. That was a memorable return visit to my high school. Mr. Hoskins, he may have died by now, but he was there, in spirit with me for my entire career.
Can you tell me how the researcher's day goes in the United States?
How research goes? Well, that's a big big question. Basic research in the United States has been very strong for many years. After World War II, the government started to provide research funds for scientists and at universities and research institutes. And as I was growing up, one of the major events was the launch of Sputnik by the Soviet Union. Mm-hmm. And that stimulated President Dwight Eisenhower at the time mm-hmm. to invest a huge amount in science education and in scientific research. And at the same time, in California, the population was growing, and the governor at the time, Pat Brown Sr., invested a lot of money in building many new campuses of the University of California. So there was a huge, in the 1960s, a huge growth in educational opportunities for middle and working class families and for science education. So that was a huge stimulus for the future intellectual and economic growth of the United States. And I rode that wave into the future. And when I got to university, there were enormous opportunities that had not existed before. For working in laboratories, I was able to spend a year in Scotland in university that was supported by scholarship from the University of California. And I got money from UCLA to work during the summer as a research student. And all of that developed, the entire enterprise around the country. And I think that is one of the major reasons why the United States has generated so many Nobel laureates compared to Europe. Europe, of course, before World War II, particularly Germany, most of the laureates came from Europe because of the enormous investment in science in Europe. But the Nazis completely destroyed Germany. The German scientists who remained left as much as possible. Einstein left in 1933, as soon as it became obvious that the anti-Semitism would end up destroying German culture. So again, World War II benefited the United States tremendously in the scientific enterprise. This enormous wealth of intellectual talent from Europe essentially all moved to the United States. And so many schools of scholarship in physics and in molecular biology came from German emigres who left and restarted their careers in the U.S. So I consider myself blessed, really. I'm not a religious person, but I feel really blessed with the opportunities that I've had as a result of just being born when I was at the right time in the right place. Not through any special talent of mine, but just being in the right place at the right time with those opportunities is really what I feel has been my major privilege.
Please describe one day. What you do in one day?
Yeah. Well, my days have changed dramatically during the course of my career. For most of my career here, my day was all entirely about the work in my lab. I would wander through the lab and ask what's new, and people would tell me, and I'd get excited, and I'd make advice. When I first started, of course, I was myself working in the lab, but as people came to my lab, I found that I had to stay in my office just to be able to raise the money and write the papers. So I unfortunately had to withdraw from the lab myself, but my interest was still primarily in the lab. And as I rose through the ranks and my work was recognized, I learned from a senior colleague who was the chairman of the department when I was hired, a wonderful man by the name of Daniel Koshland, great biochemist. I learned from him not just about doing science, I'd already learned that before, but about being a good citizen of science and a good colleague and taking responsibility for things that are not just for me, but for everyone. And so I started to take up an interest in handling the scientific literature as an editor of journals. And so I've spent many years as a journal as an editor of different journals. And so until 2012, for many years, much of my day was taken up in dealing with other people's manuscripts and making sure that good work was done elsewhere and published only good work in several journals. Then, as you may know from what you read about me, my wife had developed Parkinson's disease in the late 1990s. And for many years, it was well managed with the drugs available. But as time went on, the disease took its toll, and she started to suffer from dementia. And that became really more and more debilitating. She couldn't do things by herself. She needed help. I had to hire people to stay with her. In the night, I had to be with her just to feed her and bathe her. And as she was in deep decline, and then when she eventually died, I was approached to become a chief scientific officer of a new project, organizing an international collaborative effort on basic science of Parkinson's disease. So this had its origins in the experience I had with my wife, to some extent in my own research, but now with a really enormous contribution from a very wealthy computer scientist, Sergey Brin, who himself was from the Soviet Union. He was a co-founder of Google, and he has a family history, a genetic form of Parkinson's. He is not ill himself, but he shares a mutation with his mother who has Parkinson's. And so he's at some risk. And so he has invested enormous sums of money in science, in basic science on Parkinson's. And he decided that he wanted to make a really huge effort. And so I was basically asked to start this now, starting in 2018. And that has now become much of my day, dealing with the many different laboratories around the world, and who are organized into teams, and we're really hoping through this big collaborative effort to make discoveries that will lead to new treatments, new drugs. So I've become very interested in drug discovery. I serve as an advisor to many different biotech companies. And then this effort through this organization called Aligning Science Across Parkinson's, ASAP. That is now one of my major new things, but I also, of course, still have a laboratory and still eager to see data and discuss experiments and interpretations and writing up for publication. That's still. So I have to say I'm as busy as ever, because I get invited to give talks all over the world and talks like this, and my day is filled from morning until night. I think I get this from my mother. I enjoy keeping busy, and so I'll probably stay busy until I drop dead.
In your opinion, in the desire to study is genetically transmitted, or is another reason?
Say that again, Dennis. That my interest is to study genetics.
In your opinion, the desire to study is genetically transmitted?
Yeah. Okay, so after graduate school, I became interested in biological membranes, and I did not study with Dr. Palade, but I was greatly influenced by him. I think in my Nobel biography, I talk about a symposium where he spoke in San Diego in December of 1974. He had just received his Nobel Prize in Stockholm, and he went directly to the meeting of an organization called the American Society for Cell Biology. And I was a new member. I was not a cell biologist before that. I was a biochemist. And I had met Palade before. He was a close friend of my graduate advisor, Arthur Kornberg. And I didn't know his work that well until that lecture that he delivered, which was a version of his Nobel lecture that he'd already given in Stockholm. And it was a beautiful lecture. You never had the opportunity to hear Palade. He was a very cultured gentleman, deeply knowledgeable in many things. Really a Renaissance man. And he gave a lecture on his work, which described the organization of this process of protein traffic, secretion. And the tools that he had then available until 1974 were classical tools of microscopy and isolation of membranes from cells. The description that he gave of the sequence of events was deeply, deeply appreciated by the cell biologists in the community. He was the leading figure in membrane cell biology. But I had been trained in a more reductionist approach as a biochemist by one of the great biochemists of the 20th century, Arthur Kornberg, whose philosophy was very different. His philosophy was you have to understand the individual molecules and study them in isolation and study their catalytic mechanism. Much more detailed chemistry than what Palade had done. And so when I looked at what Palade had done and looked generally at the field of cell biology, it looked very descriptive to me and not at all mechanistic. And so looking at the image that Palade showed of this pathway, what was remarkable was that there were no molecules anywhere. I mean, there were the machinery that executed this enormous process was completely unknown. So to me, that was an opportunity. And so how to understand the mechanism. Now, James Rothman, whom I've mentioned, with whom I shared the prize, was also a biochemist by training. And his inclination, completely independently, was to use the mammalian cells as Palade had used, but to break them open and try to isolate the molecules directly by biochemical assays. But I had used that kind of logic myself as a graduate student. But since this process is so complicated, I felt that you had to have other tools to identify the proteins that were required. And for that purpose, genetics is well suited. Because if you want to study a complicated process, it's good to start by identifying the genes that are required. And the way you do that, or at least the way you used to do that, was by isolating mutations in these genes. And this you can do with living cells. But at the time, the only cells and tissues that people like Palade were using were from animals. And the genetics of an animal, or of cells cultured from an animal, is very difficult, even now. But back then, it was not feasible. But yeast is a eukaryote. That is, it's a cell that has a nucleus, and so it is fundamentally, evolutionarily connected to the lineage that leads to Homo sapiens. And so it was already known in the mid 1970s that yeast was a good model organism to study processes that occur inside of a eukaryotic cell, which is what happens with Palade's pathway. It goes on inside of a eukaryotic cell. So what I learned from that image of Palade and just my knowledge in general was that if one could use yeast to isolate mutations that block secretion, that would provide the tool needed to focus directly on the molecules of interest, and then to use biochemistry based on that genetics to discover the proteins that are required at different stages. And so Rothman and I, who are almost exact contemporaries, we proceeded quite in parallel. He was at Stanford, I was at Berkeley. He was doing biochemistry. Initially, I was doing genetics. And then when I took the genes that we discovered and was able to make biochemical reactions out of those gene products, we ended up discovering the same proteins. He discovered the proteins. I had already had mutations in those genes, and the proteins that we both isolated ended up being the same. So at first, for the years as we were in divergent, or as we were proceeding in parallel, we were rivals. We were not on good terms. We were very competitive. But at a certain moment in the late 1980s, when we realized we were working on the same thing, and then people recognized that and we started winning prizes together, we became very close friends. So we're now very close friends, and we see each other whenever I'm in New York. I see him, and whenever he's here in San Francisco, we go out to dinner. And so that has a happy resolution.
Which categories of challenges are more interesting, to prevention or to healing?
Well, prevention, well, they're both interesting, but prevention is much more powerful. If you can prevent cancer by not smoking cigarettes, that saves a lot of money and effort. Or if you can have a balanced diet instead of eating fatty foods and avoid heart disease, that's much better than having to take drugs to control cholesterol. So prevention is always better. It's better to be vaccinated than to get the virus infection to elicit an immune response. I've been exposed to COVID, but I have never become infected. I've been vaccinated 5 times. I have very good immune protection. That's much better than having to suffer the disease. And the same is true of polio and all the other viral diseases. So people who are against vaccination are simply wrong. They're just wrong. And they're putting themselves and their family members and their neighbors at risk for that stupidity. So prevention is better. Now, treating disease is, of course, important. You can't avoid cancer. We all eventually have some form of cancer. I've had several skin cancers already. It's, not possible to avoid. So obviously developing therapies for treatments is terribly important. And that's what most scientists do. Now, most scientists like me are interested in basic discovery. But when you make a basic discovery of some fundamental nature, there will be a practical application. If not immediately, eventually there will be. And so my discovery that yeast cells have the same pathway as human cells for secretion allowed the biotech industry to use yeast as a production platform for the manufacture of human recombinant insulin. This was certainly not something that would have been obvious. In fact, the first effort to make recombinant insulin was to use bacteria. But bacteria have a fundamentally different mechanism of secretion than eukaryotes. And it's very much more difficult to get insulin to be secreted by E. coli, by bacteria. And so Genentech manufactured insulin as protein aggregates inside of the bacterial cell, which have to be extracted, purified, and renatured to make native insulin. I mean, that works, but it's much more labor intensive. But if you put the gene for human insulin into yeast, you can grow it in big fermentation vats, and it just gets secreted outside of the cell. It's much easier to purify from the culture medium. So that was a big advantage, to appreciate the yeast as real eukaryote for that process. And there was nothing that I had ever anticipated doing. I mean, I was purely interested in understanding how a yeast cell works. That was my only interest. And I never had any notion that it would be practically useful, but that happens very frequently. And so that's why basic science is so important, and how it leads to drug discovery.
Let's speak about Nobel Prize. Congratulations, it's amazing. Explain please to a 15 years old student, what is this?
Yeah. Well, the Nobel Prize has a rich tradition going back to, the beginning of the 20th century. Marie Curie won 2 Nobel Prizes. The intention was and remains to reward a fundamental discovery, or the development of a new technology that has a profound impact. The prize in physiology or medicine is for a fundamental discovery of some life process. In chemistry, it's very often for the development of some new technology in chemistry. So for instance, this year's Nobel Prize in Chemistry was given to a novel approach to organic synthesis called click chemistry, which makes it much easier to build complicated organic molecules. And so that's not necessarily a fundamental discovery of nature, but it's a powerful new technology. Unfortunately, Alfred Nobel said that the prize in any given year can only go to 3 individuals. And, you know, science has grown exponentially in the last century. There are many, many more scientists working, many of them on the same thing, than there were at the beginning of the 20th century. So it becomes increasingly difficult to identify an individual or a small number of individuals to share a prize at the highest level. It becomes very difficult. And unfortunately, the committees who work very hard to make these decisions are faced with an impossible task. And the way they handle it is they sometimes make arbitrary decisions. They arbitrarily decide that so-and-so is more important than so-and-so, and another person gets left out. And every year, there are a larger number of people who are very upset than there are who are happy with the decision. Every year. So it's a problem. And the more I see of it, I mean, as thrilled as I was to receive that honor, and I continue to enjoy the benefits, the more I see of it, the more sometimes arbitrary it seems. And it's, here's an interesting thing that happened a few months ago. I was giving lectures in London, and I went out to dinner with 2 of my hosts for a seminar in London, and they had to arrange babysitters for their children. And the woman who was coming to dinner with me said that their child told the babysitter that her parents were going out with someone who won the lottery. And you know, in a way, that's true. I did. I won the lottery. It's not quite so random, but it's not too far off an analogy.
The same 15 years old boy want to know your discovery. What is this?
Yeah. Okay. So, you know, a 15-year-old boy in a gymnasium who may have been exposed to biology would know that the cell is complicated and that much of the chemistry of life is catalyzed by protein molecules, enzymes, that are a stretch of sometimes thousands of different amino acids that are linked together in a kind of a thread that folds up to create a compact protein, an enzyme sometimes. And that boy or girl might know that all of those protein molecules are coded by genes in the nucleus of the cell, and that all the protein molecules are stitched together, are made inside the cell. So in the human cell, the genes code for about 23,000 different proteins, all of which get made inside the cell. Now, most of those proteins can do what they need to do inside the cell, but a good number of them have to get out of the cell. They're hormones or molecules like insulin or antibody molecules. In fact, thousands of proteins have to have a special way to get out of a cell. Now, these proteins are mostly very water soluble. They avoid apolar, hydrophobic environments. So they can't just diffuse across the membrane that surrounds the cell. That's a barrier that would prevent a big protein molecule from escaping. So there has to be a way, a special packaging mechanism, to encapsulate proteins that are segregated away from the cytoplasm of the cell. And that's called a vesicle. And there's a process by which molecules that are going to escape the cell are threaded into a membrane that leads to a vesicle carrier, a granule of some sort. And this carrier, packet envelope, eventually carries these molecules to the perimeter of the cell, where the membrane that surrounds the carrier merges with the membrane that surrounds the cell, And what's inside the envelope is now expelled to the cell exterior without breaking the cell. And that's the pathway. That's the pathway that Palade described, and the pathway that Rothman and I were able to understand at a molecular mechanistic level. And so it's a complicated process that is very different than what bacteria do, and which evolved in the years in early evolution after we had bacteria, which evolved eventually into eukaryotic microorganisms. That was a very complex evolutionary path of trial and error to find a mechanism that was more efficient than what bacteria do to manufacture different membranes and to allow some molecules to escape from cells. So it all happened because of evolution. And at some point, the human race will discover life outside of Earth. And eventually, through maybe some spectroscopic measurements of atmospheres and exoplanets, we'll realize that the same processes have gone on completely independently in other environments. I'm certain that there is life out there. Whether there's intelligent life, who knows? But then, of course, sometimes I wonder if there's intelligent life here. But there is life on other planets. I'm certain of this. And I believe within a few decades, we'll have spectroscopic measurements of the atmospheres of planets that are light years away that will indicate molecules of biological and thus of evolutionary origin. And I wish I lived long enough to see that.
Our sponsor, Mr. Dmitry Kaminsky, is a big fan of yours. He has a similar history in family and invest energy for human longevity.
Mm-hmm.
He is want to speak. Not exist more speakers from Soviet Union who can explain. He's a speaker in international
Yes.
projects.
Yeah.
You created the Esther and Wendy Schekman. It's anything what our Academy of Science or our medical institute can help you?
Well, the chair is already fully funded, so that's done. It was I've been thinking about this for years before I won the Nobel. I'm very comfortable financially. I didn't need the money from the Nobel Prize for anything in particular. My children are well taken care of. But I thought, again, as I said earlier, I feel so strongly about public higher education. And in the US, different from Europe or Asia, in the US, there's a mix of private universities and public universities. And that it's a good mix. There's a friendly competition between those two. The private universities very often have very wealthy donors, universities like Stanford and Harvard, and they get huge sums of money from wealthy graduates. Public universities are always struggling to keep up. It's more of a challenge to get money because many wealthy people feel, well, you're a public institution, the government pays. I pay my taxes, the government pays, but the government never gives enough. So we're always struggling, even at an outstanding place like Berkeley. We're always struggling to get money for scholarship, for resources. And having an endowed chair helps a young professor have some additional funding for his or her research. And so I'd always felt that if I had the means, I would endow a chair. And when I won the Nobel, I decided to give that money to start an endowment in basic cancer biology because my mother and sister, whom I've named this after, both died of cancer. And I knew that if I gave that money, which was not enough to endow the chair, that it would stimulate other people who have more money to contribute. And that happened fairly quickly. So we were able to raise the $2 million needed for the chair. And so now one of my younger colleagues here has the Esther and Wendy chair in molecular and cell biology. And since then, I've also endowed a graduate scholarship in my wife's honor, who died of Parkinson's. And I'm now endowing a postdoctoral fellowship. My new partner and I have endowed a chair in the chemistry department at UCLA. Again, you know, I have enough money, so I'm happy to do it for this because I think these are good causes. I don't give money to private universities. They don't need me.
And my last question, the fourth and fifth scientific revolution, which challenges it proposed to solve?
The fourth and fifth scientific revolutions, which challenge would
it proposed to solve? What are the biggest challenges of the future?
The future. Well, you know, one of the disadvantages of winning a Nobel Prize is that people think you have a crystal ball and you can look into the future. I don't. So I can barely predict the present as well as let alone the future. But in general, I think the brain, the human brain, will be a mystery for decades to come. We're only just now learning basic principles of how the nerve cells in the brain are organized. And the technology is advancing so that we can now see individual nerve cells and see them actively firing to make neural connections. Even the language in systems neurosciences is beyond me. It's just completely new, heavily computational. So young scholars are now taking up very new challenges in understanding the nervous system in the brain. And that will be a major feature of life sciences for long into the future, well into the next century. Certainly beyond me. There are still other fundamental discoveries that need to be made, and the challenge is simply to make sure that the next generation has the same advantages in pursuing scholarship that I had. And although much of what we do in the life sciences now is directed to practical applications and drug discovery, my hope, and I think most people in scholarship hope, that young people will have the opportunities that I had to pursue purely basic science approaches, unconnected to any practical applications. It's, you know, it's a hard sell for the general public. How do you convince somebody who's paying taxes to pay money for some young scientist to study yeast cells? I mean, it's a hard sell, except with a certain knowledge that basic discoveries inevitably lead to unexpected practical applications. And this happens over and over, and there are so many good examples that it's, but it has to, that lesson has to be repeated to people who don't know science all the time, constantly. They have to be told about what has come from an investment in basic science. And we need correspondingly to have young people interested in doing basic science. So that's always a challenge.
Thank you very much for your time. It's a pleasure for me to speak. You are live legend because for me the time it's go. I don't understand where when. It's a pleasure.
I'm always happy to talk to you, and I apologize when I got a little emotional earlier, reflecting on my mother. So
it's family. It's
yeah, my mother died of a brain tumor at age 69. Far too young. She didn't have a chance to see her great-grandchildren. She didn't have a chance to see me win the Nobel. Yeah.
Thank you very much.
Okay, I look forward to meeting you when I finally make it to Moldova.
Thank you very much.
All right.
You. Bye-bye.
Bye.